Bismillahirrahmanirrahim. So first of all, I am thankful to the organizers for providing me the opportunity to present and to contribute in this conference. I am Dr. Muhammad Tayyab, serving as Associate Professor in the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. So the topic of today's talk is recombinant DNA technology for the production of industrially important recombinant enzymes. So I want to divide this topic into two parts. Part one, that is actually the enzymes which are important for industrial point of view. And number two, the second part is recombinant DNA technology. So let's talk about these two portions one by one. So if we talk about enzymes, so majority of enzymes are actually protein in nature. And we know actually proteins are very important biomolecules. So particularly when you are talking about enzymes, so enzymes are very important. We can see here a variety of applications. These are actually a few of examples, like these can be utilized as biocatalysts in research, in diagnostics, in food and beverage industry, in animal feed industry, pharmaceutical industry, biofuel, and detergent industry. We can have many more applications of enzymes. So here we have some of the examples of enzymes, like for example, we can see here in front of us like glucomylase, glucose oxidase, pectinase, recombinase, endonuclease, polymerase, protease, phytase, lipase, and many more. These are few of the examples of enzymes which are important for the industry. And if we talk about the production of enzymes, basically, commonly there are two strategies which are being followed for the production of enzymes. Number one, that is called fermentation technology, and number two, that is called recombinant DNA technology. In case of fermentation technology, we are going to utilize the wild type strain for the production of some special products, some enzymes. Like for example, here we can see, so we must have actually the inoculum of the wild type microbe, and we are going to provide it to the fermentation medium. And this medium is actually having various nutrients which are being utilized by the microbe for the production of intracellular or extracellular enzymes. It depends on the microbe and its ability to produce various variety of enzymes. So the enzymes which are being produced as a result of this fermentation process that can be purified and characterized later on so that you can have the final product. Whereas in case of recombinant DNA technology, that is the advanced form of technology which is being utilized nowadays for the production of recombinant enzymes. Before going towards the recombinant DNA technology, let's have a little description about a comparative analysis of fermentation technology versus recombinant DNA technology. So in case of fermentation technology, basically we are going to utilize the wild type cells for the production of enzymes. But in case of fermentation technology, if we are interested in the production of thermostable enzymes, definitely its source will be hyperthermophilic microbes. And we know that hyperthermophilic microbes, these can be grown beyond 90 degrees Celsius. And in case of fermentation technology, it requires the maintenance of 90 degrees Celsius for each batch of microbe so that it can grow and can produce desired amount of enzymes. But the thing is that Pakistan is deficient actually in fuels, in fossil fuels. So this is not a good idea to utilize fermentation technology for the production of thermostable enzymes because it will increase the production cost of the enzyme. And secondly, the yield is low. So overall, we can say that industry, as you know, that industry requires enzyme production in tons and tons, means a large quantity of enzyme is required when you're talking about the application of enzymes in industry. So it is difficult to produce these higher level of enzymes by fermentation technology at economic cost. So, uh, when you are going to produce the same enzyme by recombinant DNA technology, so you can say that we just need to grow the microbe once at its desired temperature. Once you have the microbial growth, and might be 100 ml of this microbial growth will be sufficient. So we have to utilize that micro for the isolation of genomic DNA. Once we have the genomic DNA, we can utilize it for cloning of your desired gene of interest in specific vectors, so that you can have the production of enzyme. And the important thing is that if you are going to utilize some mesophilic microbial strain for the production of as the expression host, so that can grow simply at 37 degrees Celsius. And we know in Pakistan, most of the year we are having this temperature, around this temperature. So enzyme production that is going on at 37 degrees Celsius in case of recombinant DNA technology. But the produced enzyme will have the same characteristics as was produced by the wild type strain. So this means recombinant DNA technology that is most appropriate and suitable for the production of larger quantities of enzymes. So here we have actually a brief description about the recombinant DNA technology. So I'm going to share actually the recombinant DNA technology for the production of enzymes which are required in the poultry feed industry. So there are some of the steps which are important regarding the production of recombinant enzymes. Like, for example, so first of all, we must have actually the hyperthermophile. So the hyperthermophilic strain that will be actually required for the isolation of genomic DNA. Once we have the genomic DNA, we can utilize that genomic DNA for the amplification of the gene of interest by the polymerase chain reaction. Then once we have actually the amplified PCR product, we can utilize these products. Like, for example, we need actually uh, phytase, cellulase, xylanase, and protease. These are a few of enzymes which are important for the supplementation of poultry feed. So these enzymes, once the genes has been amplified by PCR, then these can be cloned separately in the cloning and expression vectors, and the presence of gene in the vector can be confirmed by plasmid isolation and restriction analysis. So once we have confirmed that our gene of interest has been ligated in the vector, so then this recombinant vector can be utilized for the transformation of E. coli BL21 codon plus cells, for example. That is actually can serve as expression host. Then once we have the cells which are having the recombinant vector, so these cells can be utilized for the production of recombinant enzymes. And these recombinant enzymes, after the production, we can optimize actually the conditions for the production of these enzymes. And once we have the characterized enzymes, we can actually utilize these enzymes for the supplementation of poultry feed. And then ultimately by poultry trials, we can get information about the impact of these enzymes on the growth of poultry. So this was a very simple discussion about various steps important in the recombinant DNA technology for the production of recombinant enzymes. So as a conclusion, we can say that recombinant DNA technology that is actually a latest and important technology for the production, for the bulk production of your desired enzymes at economic level. So thank you. If you have some question, you are welcome. Thank you.